Hello, everybody. This is Mr. Bishop. Hello, ladies and gentlemen. Thank you for watching this evening. I know that this screencast is going up late, so if you do not get to it tonight, that's okay. A lot of you have to go back and watch the first one or second one anyway, first one as well. This is our third and maybe final screencast for our cell notes, and we will pick up with chloroplasts. It was a little bit disjointed at the end. So, just to reintroduce the chloroplast. Chloroplasts are present in plant cells. They are the energy generator of the cell. They use light energy to produce food through the biochemical process photosynthesis, and they have a double membrane. Chloroplasts are the food producers of the cell. The organelles are only found in plant cells and some protists, such as algae. Animal cells do not have chloroplasts. Chloroplasts work to convert light energy of the sun into sugars that can be used by cells. The entire process is called photosynthesis, and it all depends on the little green chlorophyll molecules in each chloroplast. Plants are the basis of all life on Earth. As we know, they are classified as the producers of the world. In the process of photosynthesis, plants create sugars and release oxygen. The oxygen released by the chloroplasts is the same oxygen you breathe every day. Mitochondria, which we'll talk about shortly, work in the opposite direction. They use oxygen in the process of releasing chemical energy from sugars. Chloroplasts contain three membrane systems that absorb light energy. The thylakoid membrane space, highly folded. It contains the thylakoids, which absorb, trap light energy. Chloroplasts capture light energy from the sun to produce the free energy stored in ATP, or adenosine triphosphate. That is a molecule, and NADPH, another molecule, through a process called photosynthesis. Chloroplasts are one of the many unique organelles in the body and are generally considered to have originated as endosymbiotic cyanobacteria. In this respect, they are similar to mitochondria. Again, we'll talk about those soon, but are found only in plants and protista. Both organelles, the chloroplast and the mitochondria, are surrounded by a double-celled composite membrane with an intermembrane space. Both have their own DNA and are involved in energy metabolism, and both have reticulations, or many foldings, filling their inner spaces. We'll hit the high points for the structure of a chloroplast. Two membranes contain and protect the inner parts of the chloroplast. They are appropriately named the outer and inner membranes. The inner membrane surrounds the stroma and the grana, stacks of thylakoids. One thylakoid stack is called a granum. Chlorophyll molecules sit on the surface of each thylakoid and capture light energy from the sun. As energy-rich molecules are created by the light-dependent reactions, they move to the stroma, where carbon can be fixed and sugars are synthesized. The stacks of thylakoid sacs are connected by stroma lamellae. The lamellae act like the skeleton of the chloroplast, keeping all of the sacs a safe distance from each other and maximizing the efficiency of the organelle. If all the thylakoids were overlapping and bunched together, there would not be an efficient way to capture the sun's energy. And here is our mitochondria. Mitochondria are known as the powerhouses of the cell. They are organelles that act like a digestive system, which takes in nutrients, breaks them down, and creates energy-rich molecules for the cell. The biochemical process of the cell are known as cellular respiration. Many of the reactions involved in cellular respiration happen in the mitochondria. Mitochondria are the working organelles that keep the cell full of energy. Mitochondria are small organelles floating free throughout the cell. Some cells have several thousand mitochondria, while others have none. If we were to think about what type of cells would have a lot of mitochondria, Muscle cells, they need a lot of energy, so they have loads of mitochondria. Neurons, cells that transmit nerve impulses in our brains and throughout our body, don't need as many. If a cell feels it is not getting enough energy to survive, more mitochondria can be created. Sometimes a mitochondria can grow larger or combine with other mitochondria. It all depends on the needs of that particular cell. So, as we said, organelle in plant and animal cells, it breaks down chemical energy for cellular use, has a double membrane. The inner membrane is highly folded, large surface area for biochemical processes to take place. Mitochondria are shaped perfectly to maximize their productivity. They are made of two membranes, similar to the chloroplast. The outer membrane covers the organelle and contains it like a skin. The inner membrane folds over many times and creates layered structures called cristae. The fluid contained in the mitochondria is called the matrix. The folding of the inner membrane increases the surface area inside the organelle. Since many of the chemical reactions happen on the inner membrane, the increased surface area creates more space for reactions to occur. If you have more space to work, you can get more work done. Similar surface area strategies are used by microvilli in your intestines. Mitochondria are special because they have their own ribosomes and DNA floating in the matrix. They are also structures called granules, which may control concentrations of ions. Cell biologists are still exploring the activity of granules within mitochondria. So mitochondria provide energy for the cell. Energy is stored in other molecules for easy access when energy is needed. So we store our energy in humans as fat, and we break down our food into simple sugars and store it as fat if it isn't immediately used. The cytoskeleton. You will find microfilaments in most cells. They are the partner of microtubules. They are long, thin, and stringy proteins, mainly actin compared to the rounder, tube-shaped microtubules. We like to say you can find them here or there, but they are everywhere in a cell. They work with microtubules to form the structure that allows a cell to hold its shape, move itself, and move its organelles, making the cytoskeleton. All of the microfilaments and microtubules combine to form the cytoskeleton of the cell. The cytoskeleton is different from cytoplasm, cytosol, which we already talked about in a previous screencast. The cytoskeleton provides structure. Cytoplasm is just a fluid. The cytoskeleton connects to every organelle and every part of the cell membrane. Think about an amoeba. All of the pieces work together so that the foot might reach out towards the food. Then lysosomes and peroxisomes are sent to begin digestion. The movement of the cell membrane, organelles, and cytoplasm is all related to the tubules and filaments. You also find microfilaments in muscle tissue. They are called myofibrils when you find them in muscles. The two proteins, myosin and actin, work together to help the muscle cells relax and contract. The two proteins need each other, and together they are called actomyosin. Combine those protein threads with some ions in the muscle cell and you get a huge contraction. The group of actomyosin contracting are called sarcomeres. All of the muscle cells work together to make a muscle contract. Microtubules, thick protein tubes. Microtubules are usually discussed with microfilaments. Although they are both proteins that help define cell structure and movement, they are very different molecules. While microfilaments are thin, microtubules are thick, strong spirals of thousands of subunits. These subunits are made of the protein called tubulin, and yes, they got their name because they look like a tube. Elements of the cytoskeleton, so we're focusing on the cytoskeleton. All of the microfilaments and microtubules combine to form the cytoskeleton of the cell. As we said before, cytoskeleton provides structure. Cytoplasm is just a fluid. The cytoskeleton connects to every organelle and every part of the cell membrane. Moving chromosomes. So microtubules, part of the cytoskeleton, have many more uses than just cell structure. They are also very important in cell division, which we'll talk a little bit more about soon. They connect to chromosomes, help them with their first split, and then move to each new daughter cell. They are part of a small pair of organelles called centrioles, which we'll talk about in a little bit. They have the specific purpose to help a cell divide. Once the cell has finished dividing, the microtubules are put to work in other places. Moving organisms. Beyond the role they play in internal cell movement, microtubules also work together to form larger structures that work on the outside of the cells. They can combine in very specific arrangements to form cilia and flagella. Cilia are little hairs you might see on the outside of a paramecium or other protists. They flap back and forth to help the cell move. Flagella are long, thick tails. They whip around and sometimes twirl, pushing the cell along. So they aid in locomotion or motility. They are composed of microtubules enclosed by the plasma membrane. These structures bend. They aid with movement, motility, locomotion, movement. Cilia are short, numerous, hair-like projections. They have a wave-like motion. And flagella are lash-like projections, whip-like motion. So you can see the two side by side. The cilia on the left in the blue and purple and green, and the flagella on the right in the yellow, blue, and green. So the cilia have that very hair-like appearance, and the flagella are longer tail-like structures. Centrioles. Cylindrical bundles of microtubules that help to separate the two sets of chromatin during cell division. So centrioles organizing chromosomes. Every animal-like cell has two small organelles called centrioles. They're there to help the cell when it comes time to divide. They are put to work in both the process of mitosis and the process of meiosis, which again we'll talk about soon. You will usually find them near the nucleus, but they cannot be seen when the cell is not dividing. And what are centrioles made of? Microtubules. So our last three organelles, the cytoskeleton, centrioles, and flagella and cilia, are all made of microtubules and microfilaments. Centriole structure. A centriole is a small set of microtubules arranged in a very specific way. There are 9 groups of microtubules. When two centrioles are found next to each other, they are usually at right angles to one another. The centrioles are found in pairs and move towards the poles, opposite ends of the nucleus, when it is time for cell division. During division, you may also see groups of threads attached to the centrioles. Those threads are called the mitotic spindle. Relaxing when there's no work. We already mentioned that you would find centrioles near the nucleus. You will not see well-defined centrioles when the cell is not dividing. You will see a condensed and darker area of the cytoplasm called the centrosome. When the time comes for the cell division, the centrioles will appear and move to opposite ends of the nucleus. During division, you will see 4 centrioles. One pair moves in each direction. Interphase is the time when the cell is at rest. When it comes time for a cell to divide, the centrioles duplicate. During prophase, the centrioles move to the opposite ends of the nucleus, and a mitotic spindle of threads begin to appear. Those threads then connect to the now apparent chromosomes. During anaphase, the chromosomes are split and pulled towards each centriole. Once the entire cell begins to split in telophase, the chromosomes begin to unravel and new nuclear envelopes begin to appear. The centrioles have done their job at that point. So, that concludes our notes for organelles in Chapter 7. We may have another screencast or two to do some review and discuss a few more things, but I want to thank those of you who have taken the time to watch each of these. I think it really shows with your enthusiasm in class that these are helping you. And for those of you who have yet to totally buy in, I hope you are ready to commit, because we'll be using the screencasts as our mechanism for note delivery going forward. I think our our activities in class are more important than note taking, and thank you all. Have a good night, and I'll see you in the morning.